Welcome to the Division of Education, Innovation and Energy e-learning program. I am Miss Jamilla Bacchus, your facilitator for CAPE Biology, and today we are looking specifically at Unit 2 Biology, and we're looking at Module 1. We want to review photosynthesis. If you would recall, that would have been one of the first topics we would have done in Module 1. And we want to look at the importance of photosynthesis, but we are going through very quickly because this is a review. You may have done it before in school, and so we're looking at the overall process of photosynthesis and all that it entails. Good. So what do we see here? I can ask the question, what do all these things have in common? You have a newspaper, we have plastic bottles, we have food in all its many variety, and we have coal. Yes, coal that you burn, that they will be using as a source of energy, whether in trains or, to make your barbecue and so on. Coal. What do all of them have in common? Yes, the answer is photosynthesis. You would agree that photosynthesis would be the way in which energy would have been trapped initially, in this case by green plants, and these green plants can either form different products that can form things like paper, or they can be buried and become, parts of things like crude oil, which will be within the depths of the oceans and the soil and so on. And these can be drilled up, and the crude oil can be made to manufacture things like bottles, or like paints, or like plastics that you have in your furniture and so on. So photosynthesis, of course, we know the obvious relationship with plants. The energy that is captured can be used and is formed in all the different parts of a plant. And coal, again, this would have been plants that may have been buried below the earth, and the pressure and the heat within the earth can actually form coal. So it's really plants that would have been preserved here in this format, and so it contains a lot of energy. So photosynthesis is the link between all of these things. What would be our objectives today? At the end of this lesson, we want to be able to recall the structures the locations, and the metabolic reactions involved in the process of photosynthesis. We also want to review the overall process of light and dark stages of photosynthesis. I know some of you, you would have heard light dependent and light independent, or light and dark stages. We'll clarify that. We also want to be able to appreciate the importance of photosynthesis to all ecosystems and the sustenance of life. Can we do without photosynthesis? Can we survive without photosynthesis? Let's look at that. Of course, we want to be able to answer CAPE-type questions at the end of this lesson. So, what is this? We are seeing here a leaf, and the leaf, we know, is the principal organ that is responsible for trapping light energy. So we know the sun is the source of all energy on the earth. Okay? The sun gives out a lot of energy. How can we get that energy? Can you as an individual go out into the sun and just capture that energy? Can you open up your mouth and eat that sun's energy? No. In spite of your best effort, you cannot capture that energy yourself. Good? All the energy that comes from the sun, it has to be captured by an autotrope. And the autotrope that is mainly responsible for capturing that sun's energy, that solar energy, is going to be the leaf. And the leaf is a unique structure which makes it very efficient in capturing that energy. You see in here, the leaf has, it takes in water. So the water which would have come from the roots of the plant will now be in the leaf, and you will see water is very important in the process of photosynthesis. The leaf also has chloroplast. That is what gives it its green color. But note, it's not only the chlorophyll which will be present. You have other types of pigments, and we'll look at that. And so you have this leaf which has special features which allows it to be efficient. What are we trying to do? We are trying to capture that energy. And energy simply can be transferred from one form to the next. We have potential energy and we have kinetic energy. Okay? The potential energy is the energy that something will have stored. Good? It has the potential to do work. And the kinetic energy is the energy that something would have because of its movement, because of its transition, right? And so potential energy can be converted to kinetic energy. You have stored mechanical energy, you have nuclear energy, you have chemical energy. So when we eat food, when you eat that piece of bread, when you eat that piece of chicken, guess what? It has energy within it. It has chemical energy within it. You have gravitational energy, energy you have with respect to your position in terms of gravity. You have electrical energy. You have light energy, again, coming from the sun. You have heat energy. So when you feel that you go near to a fire and that heat is coming out, or when you simply hold someone and you feel their body, you feel the heat, because they are actually releasing heat energy as they live. That's why a dead body feels cold, because it is no longer releasing energy. And then you have movement, which will allow energy to be transferred. Let's look a little deeper at this leaf. Here you can see the structure of a whole leaf. And what are the features of this leaf which makes it so efficient in carrying out the purpose that it was intended, which was photosynthesis, trapping that light energy? You can see from just looking at it from a surface view that this leaf has a very broad surface. It has a very wide lamina. And so that allows it to be able to capture a lot of light. And the thing about this leaf is that, you know, according to where it is, the size of this lamina can change. Because also on this leaf, you have below it holes called stomata, which play an important role in bringing in gases or releasing gases that will be needed or produced. Another thing about this leaf, it is very thin, T-H-I-N. Okay? And the fact that it is thin means that as the light passes through, you can still have another leaf that is below. I know it's not the best drawing of a leaf, right? A leaf that is below that can capture that energy. So as the energy goes through, the fact that this is thin, it can percolate through the leaf and get to the leaf below, and it can still go down. So even though a tree is very large and you have several hundred leaves, you will still find the leaves at the lower level will be able to get some of that light energy. Another thing about the leaf, you have this structure here, this structure at the end here. Now this structure is called the petiole. Petiole. And the petiole connects the leaf to the stalk. It connects it to the rest of the tree. Now this seems very simple, but this has a key role, because you will realize, you know, at different times of the year, if you really watch the leaves properly, they actually turn. Because the leaves are arranged so that all the leaves in a tree can actually face the sun. And so at different times of the year, because the sun changes position, the petiole allows the leaf to be able to change position so that it is always facing the sun. So this plays a very important role in these plants. Okay? Let's look at the cross section of a leaf. It's actually a 3D because we are seeing greater depth, right? And here at the surface, you have the cuticle. This cuticle is very waxy. It's made of wax. The wax that you will have, you know, on apples, on candles, and so on. On the surface of the leaf has this wax. And according to where the plant is located, the thickness of this wax will differ. So a plant that is in a xerophytic environment, a very hot environment, will tend to have a thicker cuticle, because what this cuticle does is it prevents the loss of water through evaporation. Okay, so if you're in a hot environment, you will want to prevent too much water being lost, because you need the water for the process of photosynthesis. Other parts of the leaf, you will realize this area here is the epidermis. This is the upper epidermis. It has no chloroplast, and it plays a very important role in protection and facilitating the passage of the light rays through. This here is a key area. This is the palisade mesophyll cells, and it is here that most of the photosynthesis takes place. If you realize the way in which these cells are oriented, they have this longitudinal side like this, as opposed to being like this. Because when you have the chloroplast arranged, as you see here all along, if you have this arrangement with the chloroplast all along, as you can see here, if light is coming through, then as the light comes through, it's captured by some chloroplast here, but you can still have the light continuing down, and so you can maximize the way in which the light will be captured, as opposed to here, if it is like this, then it passes through here, and then it's gone. So this arrangement of the palisade mesophyll allows these structures to be able to maximize how much light they can capture as the light passes through. You also have the spongy mesophyll cells, which also play a role in capturing some of the light energy. However, their main role is really involved in water, movement of water, and storage of gases and so on. So they are not the principal part for photosynthesis, but they do carry it out. You also see in this location here, this is the xylem. And of course, we know xylem is very important in transporting the water and minerals so that the leaf can get the water for the process of photosynthesis, and it can also have the minerals which are needed to form some of the molecules that will be involved in the product in photosynthesis. You also have the phloem in this location, and the phloem plays a very important role in moving the assimilates, moving the products of photosynthesis away. Lastly, you have this layer here, which is the lower epidermal layer. And like the upper epidermal, most of the cells do not have chloroplasts. But look at this location here. Here you are seeing an opening called a stomata. Let me just clear this a little to give you a better view. So here you have a stomata. This is like an opening or pore on the surface, so it allows gases to move up and get into the leaf. It also would allow the loss of excess water. And these cells, because they play a very important role in they open and they close to allow the entire thing to close, like at night, or if you're losing too much water, they close. They need energy, and so these have chloroplast within them, which will be able to capture some of the energy and have enough energy right there for itself. However, the rest of the lower epidermis contain no chloroplast. We continue right after this. Let's zero into the chloroplast. As we know, these are the actual units which play an important role in trapping the light energy. And if we look at this structure, we see some unique features. Remember, we would have looked at the chloroplast in Unit 1, and we would have had the basic structure. So here now, we want to appreciate what is happening, where photosynthesis is happening within this structure. So you have an outer layer, okay? And so this outer layer, and then you have an inner membrane, an inner layer. So you have a double membrane. And this is very important. This, remember when we looked at the endosymbiont theory, we would have hypothesized that there are two layers because of the fact that this structure was once an independent organism living on its own and was actually taken in by another. So Unit 1 would have dealt with that. You have now the stroma, which is this watery area here, which contains a lot of enzymes and a lot of hydrogen ions and so on. And you have the lamellae, and you have the thylakoid. So the thylakoid, and we have them stacked to form a granum. So these structures, the thylakoid, plays a very important role in the actual process of photosynthesis. So these are like, think of it like coins that are stacked on top of each other. If we should cut this down, what we will see is that the thylakoids are like that. And this would be the external structure. So this would be their membrane. And inside of here would be a space called the lumen of the thylakoid. Right? So the lumen of the thylakoid, and this we have to appreciate because what is happening is that you constantly have hydrogen ions moving between here, and that is responsible for the production of energy. You also have the lamellae, which is within the stroma, which connects the various granum. And so you have interconnectivity within this structure. Very, very beautiful structure. And I would have had some of my past students build these things. And I would suggest you try to build it too, because it really gives you a hands-on and appreciation of what is happening. Note, the stroma is a type of matrix. I made mention of this because very often students confuse the mitochondria and the chloroplast. The mitochondria has a matrix, we call it the matrix, and the chloroplast has a matrix, we call it the stroma. So don't confuse both of them, please. Right? So within these thylakoid membranes, what we have are photosynthetic pigments. Okay, we have these little packets which can actually trap the light. So remember, the thylakoid membranes contain these photosystems, which are made up of certain types of different pigments. We have chlorophyll and what we call the accessory pigments, which together trap light. And that's why I said this is the unique structure which does the massive role of capturing that light. Right? You have here chlorophyll A, which is the main pigment for trapping the light. And this is actually the molecule that is found in what we call the reaction center of the photosystem. And this is found in all photosynthetic eukaryotes and cyanobacteria, essential for photosynthesis in these organisms. Then you have chlorophyll B, found in vascular plants, bryophytes, green algae, and eulinoid algae. It is an accessory pigment. So an accessory, we all know what an accessory is. something that assists, something that will work in conjunction with it. It's not the main thing. So chlorophyll B is an accessory pigment, very, very important, we will see. You also have another accessory pigment, the carotenoids, and you have different colors. These are the ones you will see more or less in autumn when you see the chlorophyll, the main one being broken down, the green, you will see the green color being broken down, and then you will see the reds and the yellows and the orange and the browns. Those are different types of carotenoids and other pigments. Even in the sea, you will see different color seaweeds. And because different pigments are there, accessory pigments, that play an important role in trapping some of that light energy. You have the xanthophylls, which are another type of pigment. So, what do we have here? This is the electromagnetic spectrum. What this shows is the different wavelengths of light and the amount of energy that will be found in different waves. So you go from the gamma rays to the radio waves. And you will see the gamma rays, these rays contain more energy. You will see they have higher frequency, but they have shorter wavelength. And as you go along, you see the increases in the wavelength. The wavelength, it will go from here to here is one wave. Okay, and from here to here is another wave. So you see the increases in the wavelength, and you see the decreases in the frequency of the waves, right? So the frequency of the waves is decreasing. So what you are seeing here is that energy, according to the type of rays, they will have different levels of energy. Now we will see these types of waves. Within here, you have within this small area here, you have what you call visible light. So we will see this because if you know, how do you know something is a certain color? You can see that color. So if I look, I'm seeing a red chair, I'm seeing a green leaf. Now the color that we see is the light that is reflected. What we don't see is what will be absorbed. So you will see between here and here, 400 nanometers to 700 nanometers, we have what you call visible light. And of course, again, at this end, because it's closer to this end, the light rays on this end has more energy, and as we go down, this has less energy. So when the leaf captures energy, according to the rays it captures, will determine how it will be able to convert that ray into chemicals. So you find light that would be of a higher frequency and lower wavelength will have more energy than the ones that will be of a longer wavelength and a lower frequency. Good? So this is visible light. We all know about Roy Breath, and we should be able to recall that from our lower forms. Good. So what are we looking at here? This is the absorption spectrum. So as we saw a little while ago, we saw the entire spectrum, and now we were looking at visible light. So this is showing us how green plants are able to capture the light rays, and it's showing the different pigments and where they will capture most of their energy. You will note that chlorophyll A, which we say is the principal pigment, is actually capturing a lot of the light rays close to the 400, and it also captures close to the 700. You will find during this point here, between here and here, you will not find chlorophyll A trapping a lot. But what if light comes and it has all these different wavelengths in it? Will the wavelengths within here be lost, be not, captured? The answer is no. Because of the accessory pigments, they will fall in other areas where chlorophyll A may not fall. And so the combined capturing of the light rays by all the different pigments will now allow the plant to get an increased amount of energy. And that is the work of the accessory pigments. They actually, they enhance the chlorophyll A in capturing light. So even though light will fall, and you have several different wavelengths of light within the photon of light that is captured by the plant is able to maximize. So this is the absorption spectrum. And what we have found is that there is a correlation between the absorption spectrum and the action spectrum. The action spectrum has to do with how well the plant is able to now take that energy it has captured and convert it into products. Convert it into glucose, convert it into viable products within it. And so the action spectrum, we have seen where you had the high level of absorption also shows where you have a high level of action, the high level of producing the products. So both at close to 400 and close to 700, we are able to have a very high production of things that you can use. As I said, within this region here, you will notice that this region more or less is green, and you will notice leaves are green. That is because those wavelengths of light are not being absorbed. They are being reflected. Good? So the energy, it's being reflected in the form of light energy, and we see green leaves because that light is not being captured. I am certain that all of this is recalled. I'm certain that you would have done this previously, but it's just to go over it to give you a chance to marinate on it and get a deeper understanding of it so that you can go into exam being very confident of your knowledge. So here we see the photosystems. Take a break. Right, so we spoke about the photosystems. Remember we said in the thylakoid membranes, we have these pigments. And these pigments are arranged in these clusters called photosystems. So the photosystems are really different pigments, molecules of these different pigments. And these things, they work together to capture light. So you capture light, which is striking the plant, striking the leaf. And these are within the thylakoids, all right, within the chloroplast. So when the chloroplast receives it, these molecules will pick up this light. And what do? Now, all of them will collectively move the light until it is collected in one place. So they are acting in unison to bring the light rays and the energy within that light rays to one place, which we call the reaction center. Now within this reaction center is where you will have chlorophyll A. So within this whole antenna system, we call it an antenna system, you have several different types of pigments. So you may have some chlorophyll A, some chlorophyll B, some carotenoids inside of here, but they all will simply capture the energy and pass it on to each other until eventually they allow this energy to be captured in the reaction center. What will happen now, this energy will cause this molecule to elevate an electron to a higher energy level. And this electron can now literally jump out of these reaction centers. As we go along, we're going to watch at a little video, and we're going to watch at what we call the Z scheme, or the the cyclic and non-cyclic process of photophosphorylation, where this energy that is captured in the reaction center is then passed on, and this is what is responsible for allowing the production of ATP, which is the small carrier molecule which, you know, allows work to be done within your body. So the purpose of the photosystem is to collect energy over a broad range of wavelengths and concentrate it to one molecule called a reaction center, which uses the energy to pass one of its electrons onto phaophytin, then a series of electron acceptors. So let's look at a video. And the interesting thing about this video is that it was produced by two of my past students. So we would have had a project where they had to produce a series of things related to photosynthesis. And one such thing is that they were supposed to explain the non-cyclic photophosphorylation and use, you know, some kind of technology to do it. And they came up with this. I'm so proud of these two students, as I am of all of my students. And you have Miss Selena Campbell and Miss Anisha Crooks, both of who are tertiary level at this time. So we look at this video now.
Hi, today we are going to learn about the non-cyclic reaction in the light-dependent stage of photosynthesis. Photosynthesis is where plants use energy from sunlight to produce glucose and oxygen from raw materials of carbon dioxide and water. This reaction takes place in the chloroplast in two stages. Light reaction, which occurs in the thylakoids, and dark reaction in the stroma. The membrane of the thylakoid is like any other. It is a bilayer consisted of different proteins. As you can see, there are proteins embedded in the bilayer. These different proteins are Photosystem 2, H+ ion pump, Photosystem 1, and ATPase. A photosystem is a biochemical mechanism in plants which light energy is absorbed by chlorophyll in photosynthesis. This membrane is the barrier between the stroma and the thylakoid lumen. At photosystem 2, light energy hitting the membrane excites electrons to a higher state and is carried through an electron transport chain. The electrons lost by photosystem 2 is replaced by a reaction called photolysis. Photolysis is the splitting of water molecules which produces H+ ions, oxygen gas, and electrons. As the electrons pass through the electron transport chain, they lose energy, which is used to pump the H+ ions from the thylakoid into the stroma, creating a concentration gradient. The gradient powers a protein called ATP synthase, which allows photophosphorylation of ADP to ATP. The electrons leave the electron transport chain with low energy as they approach photosystem 1. Light enters photosystem 1 simultaneously with photosystem 2. So these low energy electrons are now re-energized and are carried through another electron transport chain. They are now used to reduce NADP+ an electron carrier to NADPH. ATP and NADPH are released in the stroma.